So I only have 20 minutes, and one can't possibly do justice to this topic in that time, so I'm going to fly through some information and some data for you. So I'm a neurologist and a neurobiologist, and what I do for a living and try to affect is try to design and conduct clinical trials. And I was a clinical neurologist, took care of these patients for a number of years. And I'm going to talk about the larger landscape of neurodegenerative diseases and the unmet need. And as you can see here, and this is somewhat dated information, but these trends are actually just increasing, that neurologic diseases is actually going to become the leading cause of disability and the second leading cause of death. And so it's a really large unmet medical need, which really, because of the chronic nature of many of these conditions, induces a great deal of disability in the patients, which impacts society, both the patient level of function, but also economically. And degenerative diseases such as Alzheimer's and Parkinson's disease are significant and disproportionately affecting the population. So I can provide a copy of some of these slides to you if you're interested after the study. And so when you look at disability life-adjusted years, you see that neurologic diseases, as age increases, that really has a bigger and bigger impact. That's really growing exponentially. And after the age of 50, this is really a very significant impact on a population basis. And so when I think about neurodegenerative diseases, I think about symptomatic benefit and then disease progression impact. You know, a lot of the focus has been on trying to develop disease-modifying therapies, you know, but from day to day in the patient's life, don't go by or don't surpass symptomatic treatments. Those can really help patients as well. So we really need to think about both of those approaches. And this, of course, has a huge economic impact, and it's going to grow as we age and the population ages. And this is just some detail and some of the dramatic impact this is going to have as it increases over the next 30 years. Neurologic drug development has been very challenging. It's a tough area to try to be successful in. And this is just a list of different types of therapeutic areas and the likelihood of being positive in phase 1 to phase 2 to phase 3. And unfortunately, you can see that neurology is down near the bottom of this list, okay? And so it's been difficult. Now that's changing. I think as we've developed biomarkers, we've had better pathogenetic insight into disease mechanisms. I think we're making progress. It's been incremental, but I think it's been accelerating over the last, let's say, 10 years. And in the pharmaceutical industry, they're nothing if not bellwethers of the direction of the moment. In other words, if there's a big success in a particular area, pharmaceutical industries will pile on and think, oh, we'll go into that area because someone's had success. If there's a big disaster in an area, they'll say, no, we're going to get out of neurodegenerative diseases. And so that will happen for 3 or 4 years. Then someone will have a success, and the same company will say, no, we're going back into it. So that's highly inefficient, but that's the way the world exists, and that's basically what we have to deal with. So I think as neurology, some successes maybe occur that this type of in and out and fluctuation, you know, may be mitigated. I think it takes an investment in time and effort. The brain is in a hard box. It's hard to get a hold of it in a clinical trial, a sample of it. And so it takes a bit of investment of time and effort to actually make a difference. Now there have been some recent very nice positive results. Karuna, local company, with KarXT was a big advance, I think, in schizophrenia therapeutics. And there have been 3 different amyloid antibodies that have been positive. And that's not going to be the answer to Alzheimer's disease, but I think it's a start to an answer to Alzheimer's disease. And so this is, I think, galvanized interest again in neuroscience. And I see in pharmaceutical companies, people are now saying, oh, yeah, we should go back into neuroscience. So, you know, as of, you know, today's weather report, the next few weeks, yes, okay, they're interested. So let's see what happens. But irrespective of all that, this first group of slides is making the point that there's a big clinical need. Okay. So why is this gap? Why have we had this difficulty in being, having not great success and being down at the bottom of the list? Okay. So this is my take on it. And I think with psychiatric conditions, there have been better therapies because psychiatric conditions are characterized as difference in activity, okay? It's circuit activity differences. Well, what are those, what subserves those circuits? Neurotransmitters. Okay, so we can modulate neurotransmitters and have an effect on psychiatric disease. Virtually all psychiatric medications are aimed at modulating neurotransmitters. Well, if you look at neurologic conditions, many of them are due to faulty wiring, okay? You know, it's a lot easier to put a new fuse in your house than to rewire it, okay? And so if a circuit is burned out in a neurologic condition, that's a bigger challenge to try to rectify than if there's alteration in activity. So that's one of the ways I think about this, and one of the ways that I think makes it more difficult for neurologic conditions to be treated versus psychiatric conditions. And you know, there have been some neurologic diseases that have had big inroads in treatment, multiple sclerosis. But multiple sclerosis is actually not a neurologic disease at all, okay? It's a systemic inflammatory disease that happens to attack the nervous system. And most of the therapies for MS are to dampen down the inflammatory condition. Remyelination, which is repair of the wiring fault in the brain, there are no drugs that induce remyelination. So that's the next horizon, for example, in MS, is how can we repair the damage that this inflammatory condition has wrought, because now we can control the inflammation in MS quite effectively, actually, okay? The other problem in neurology is we're 20 years behind oncology. One of the reasons I went into neurology is I looked at the neurology textbook and it was this thick and the index was this thick of all different diseases. And that just attracted me because I'm just interested in information and facts. But the problem is that that list of diagnoses is really empirical observations. And now we know that, for example, in frontotemporal dementia, a neurologic degenerative diseases, you can have 5 different phenotypes with exactly the same mutation. So it doesn't make any sense to treat these different phenotypes with different drugs. We should be treating the mutation. We should be treating the biology. And so this is where cancer is so far ahead of neurology. You know, if you go to an oncologist and they say, well, you have bladder cancer, you have kidney cancer, you should walk out the door, all right? So what you should hear from the oncologist is you have bladder cancer, and this is the molecular profile of your tumor, okay? I know this oncogene is turned on, this kinase is turned on, so I'm going to treat you first with drug A, then drug B, then drug C. It's a logical approach to treating the disease. We don't do that in neurology yet, but I think the future holds some hope for that we are actually understanding the mechanism of these diseases so we can use maybe, it's aspirational, a guided missile versus a blunderbuss therapy, okay, in the future. And there's a whole list of other advances. Biomarkers right now is revolutionizing selection of patients for Alzheimer's disease and determining whether a drug will have a benefit in Alzheimer's disease. Greater selectivity in drug development candidates. When I first started in drug development, many drugs failed for safety, bioavailability problems, PK. That doesn't occur much anymore, okay? We're much better at picking good drug candidates. And using imaging and other biomarkers, I think, are also an advancement. And the regulatory climate understands this. I'm not going to go into this in any detail, but I think that they've started to respond to that. And I've seen a difference in the temperament of my interactions with the FDA and the EMA and all the other regulatory authorities I interact with, which I do all the time. Okay, and so this is basically what I've said in the first half of the trial is that there's a growing clinical burden. We need to focus both on symptomatic as well as disease-modifying therapies, and there are many more tools now that will help us do this. So let's drop back 120 years, okay, to Alzheimer's disease. This is Dr. Alzheimer, Lois Alzheimer. This is the first documented patient with Alzheimer's disease. We can't, of course, name a particular patient's name in today's environment, but this is published 120 years ago, and this is Auguste Dieter. And she had a very unusual case of Alzheimer's disease, manifest by paranoia, aphasia, language problem, progressive sleep problems, behavior, and then some more typical symptoms, memory disturbance and aggression late in the disease. And he described the two genetic or pathologic abnormalities in Alzheimer's disease. That's the so-called amyloid plaque, or smudge, if you will, and the tangle, which is both an intracellular and then eventually an extracellular deposit of tau protein. And Alzheimer's disease occurs in a cascade, and I think this is now just starting to be recognized may have a dramatic impact on which therapies work when in the disease. That amyloid is probably one of the earlier pathologic events that occur, and that occurs even well before the patient is symptomatic. Anti-amyloid antibodies are most likely to be effective very early in the course of the disease in the MCI phase. And so people are moving earlier and earlier with these approved antibodies to determine whether they can be effective. However, we can't start treating patients the day they're born, okay, number one. Number two, if you go to patients with minimal clinical impairment, cognitive impairment, they don't have a clinical deficit. They have some detectable memory deficit. 40% of those patients never go on to get Alzheimer's disease. So that the 80-year-old gentleman walking down the street doing quadratic equations in his head and contemplating Wittgenstein might have a brain full of amyloid, okay? So are you gonna treat that patient with an invasive therapy that costs $30,000 a year that causes side effects? Maybe not. So this needs to be figured out. Which of those patients are actually going to go on to develop disease? Now we can characterize the removal of amyloid from the brain by the antibodies by imaging. And typically what I'm showing here is a progression from a normal individual all the way in the red is the amyloid deposit to the later stages of the disease. And what we use is a cutoff around 20 or 25 in this scale called centiloid to sort of say that this is now negative. This is positive, this is negative. And so if we look at some of the clinical trials with monoclonal antibodies to amyloid, you can see some of them get down into this negative range, some of them don't. So lecanemab and donanemab actually get down to this clearance of amyloid measure range. The two aducanumab studies, one was positive, one was negative. This is the negative one, there's a positive one, okay? And so now people think, okay, we need to get to a threshold of amyloid removal in order to have benefit. And basically, if you don't virtually completely remove amyloid for a period of time, you're not going to have benefit. So this is a cartoon or diagram that I drafted showing that over 18 months, this is the placebo group, this is the anti-amyloid antibody group, this is the benefit. Okay? In purple is the unmet medical need, okay? Ideally, we want to halt the disease, but we can't. We can just slow it. This difference is about a half a point on this CDR sum of box scale. That's the clinical difference after a year and a half of treatment. Okay, if you look at the literature and you ask, well, what's the clinically significant difference in the CDR sum of boxes? It's actually about 1.1 to 2. This is 0.45, okay? So these therapies have been approved by showing that the difference, which is not clinically significant, but statistically significant, because you're doing trials of 1,700 patients, okay? You do a large enough trial, you can prove that a tiny difference is statistically significant, but not clinically significant. Now, I'm not saying that the anti-amyloid antibodies aren't useful, they are. But they're not going to be the answer, okay? This is the effect if you completely clear amyloid from the brain. And so there are new programs that are trying to increase the ability of the antibody to get into the central nervous system to have more comprehensive and rapid clearance of amyloid. I don't have time to go in these, but most of these are focused on linking an antibody to a targeted form of amyloid to a transferrin antibody, receptor antibody. Transferrin is a molecule that your body uses to get iron across the blood-brain barrier. And so what you can do, like a Trojan horse, is you can couple the antibody to transferrin receptor, get that through the blood-brain barrier. And so what that does is that requires a lower dose, less frequent administration, more comprehensive clearance, rapid clearance, and a lower incidence of these side effects. So I think even though these antibodies may not have any greater therapeutic benefit, they're going to be better tolerated by patients, you'll be able to give them less frequently, and so I think they'll be in advance. And virtually every company working in Alzheimer's disease has a program. Now finally, ending up with where is drug development right now in Alzheimer's disease? So Howard Phillott, who's the founder and the CSO of the Alzheimer's Drug Discovery Fund, said that 5 to 10 years ago, 75% of the drugs were targeting amyloid or tau, and 25% targeted other mechanisms. Now that ratio has flipped. And that's because the understanding of the underlying pathogenetic mechanisms in Alzheimer's disease has gotten better, and because people have recognized that, if this is the therapeutic benefit we can see with comprehensive clearance of amyloid, we have a lot of work to do, and it's probably not going to come from anti-amyloid therapies, okay? It's going to come from other areas. So I refer you to this article that Jeff Cummings has written in Translational Research and Clinical Investigation 2024. It's an open access article. It's a very nice summary of where we are in Alzheimer's disease drug development. And this is basically Jeff's group's take on all of these mechanisms, the distribution in different areas of disease approaches to helping patients with Alzheimer's disease. Disease-modifying small molecules, disease-modifying biologic interventions, cognitive enhancers, and symptomatic benefit. And these are some of the areas that are most actively being explored. As you can see, there's now a greater diversity of these areas. And if you look at the stage of development, you can see that amyloid and tau, yes, there's still a lot of activity, but inflammation, neurotransmitter receptor modulation, metabolism, bioenergetics, proteinopathies. So finally, we're getting a more diverse approach to these diseases. This is a snapshot of the drugs that are in phase 3. And as you can see here, you've got 22% of them being amyloid and 3% tau. And if you look earlier, back to phase 2, what you can see now is that there's 12% in amyloid. So the number of earlier stage projects that are focusing exclusively on amyloid is going down. We're getting greater diversity, different approaches to the disease, and I think that will be very helpful. This is just a selection of some of those drugs that are in phase 3. There are approximately 40 programs in phase 3. You can look at Jeff's paper, and there'll be some detail of that. And as you can see, some of these drugs are disease modifying. Some of them are trying to be symptomatic benefit for patients. And they have a variety of mechanisms, okay? So I think my message is eventually, it is one of optimism, because I think we're diversifying our approach to this disease, recognizing, as Churchill said about the end of the Battle of Britain, he was asked, well, is this the end? He says, no, it's not the end. It's not even the beginning of the end, but it may be the end of the beginning, okay? And so where I think we are right now is I think we're at the end of the beginning of treating Alzheimer's disease. And we've got to, every chronic disease that I treated as a physician taking care of patients, I use more than one drug. I know of no chronic disease that is well treated with a single drug. And I know of no chronic disease that is amenable to a set combination throughout the entire course of the disease. It's going to require drug A, then a little bit of drug B, and dropping A and adding C and D, and then maybe later adding E and dropping B. And so that's just the reality of treating chronic disease. And so there's a big unmet need. I think we're transitioning to a more rational era of drug development for a variety of reasons, and combination of drugs, I think, will be the answer. And I'm over my time. Thank you.